I remember talking about this slide. Is that correct?
Yeah,
All right, thank you. So let's go on to chapter 6. Did we start chapter 6? Although I should have a marking on this page, so.
I think we started this.
Yeah, I've got that we were talking about enrichment media. Yeah, I remember talking about um, a phenol degrading organism in the soil. So we were talking about different types of media. The last one we talked about was an enrichment media. It is selective, but you use it to enrich for bacteria of interest. And this slide is showing you the different types of media that we have available to us, such as chemically defined media, where everything, all the chemicals are known in the media. Let me make sure I'm doing everything correct. Okay, that looks like I'm recording this. I'm sharing my screen. Anyways, chemically defined media, complex media, we don't know what all the different molecules are in a complex media. It has usually ground up beef or ground up soybean, something like that. And what the exact molecules are in the media, we just don't know what it is. And it'll vary from batch to batch because if a cow's eating one plant in like Colorado and it's eating a different plants in Oregon or Washington, then it'll be a little different. There's also reducing media. We use it to grow obligate anaerobes. There's selective media. It suppresses some microorganisms, encourages the growth of other microorganisms, and it has a chemical in it that acts as a selective agent. A differential media allows us to see differences between different types of organisms. And then an enrichment media is selective, but we use it to increase in numbers the microbes of interest to bring them up to detectable levels. Let's move. When we're talking about pure cultures, you should realize that pure culture is one that has only one species of bacteria growing in it. Louis Pasteur came up with the concept of a pure culture, and he came up with a way of obtaining a pure culture. However, that way of obtaining a pure culture was not a very easy way to obtain it. And what he did was he made different dilutions and then plated them out where he had 10 replica tubes of each dilution. And then he looked at the dilution that only gave one tube that was positive and 9 tubes had no growth in it. And he correctly assumed that the tube that was the one tube and there's 9 tubes that had no growth in it was started by one or very few cells. And so it most likely was going to be a pure culture. I don't know the percentage of that. It probably wasn't 100% the case, meaning occasionally there would be a tube that was started by 2 or more cells, in which case it was not a pure culture. But the point is, most of them were, and it's probably about 70% or over that, maybe even higher because this is a liquid culture that he's working with. When you're working with solid culture, the cells tend to stick together. And so the clumping is a little more frequent. Anyways, it was a complex method. It involved setting up many different tubes, 10 tubes for each dilution. You had to make a different dilution and a large number of dilutions, and then all grow them up. So when he was doing this, he probably had something close to 100 different tubes. Is that right? Yeah, close to it, something like that. And then he was just looking for one tube to try and get the pure culture. Louis Pasteur developed not Louis Pasteur, Dr. Koch developed a much easier way of obtaining a pure culture. And he just streaked out on a solid media, getting isolated colonies. And then you take from the first isolated colony and you streak it out a second time on a solid media. You usually use auger plates. He didn't initially use auger plates in the very beginning. This is just a story for your own amusement. He was streaking it out on a potato. He cut a potato. I'm assuming it was a raw potato, but it might have been a cooked potato, and then streaked out the bacteria on the potato. It wasn't a very good way to streak out your cultures, but that's the first way he did it. And then he did gelatin plates, and gelatin wasn't a very good culture media either because some of the bacteria dissolved the gelatin. And then you got liquid gelatin and all your colonies merged together in the liquid. And it was actually the wife of one of his assistants who suggested using agar. Agar is an agent that is used in cooking or baking, and it's just used to thicken whatever you're cooking or baking. And she suggested agar, and he tried it, and he found that no microorganisms that he was using could digest agar. And we still use agar today because it's so good and the microbes don't digest it. Anyways, he got a colony. A colony is a population of cells arising from a single cell or a single spore or a group of attached cells, meaning more than one cell. And therefore, when you streak it out, you shouldn't say how many colony forming units you got to grow on a plate, and not colony forming unit, how many cells were derived on the plate, because each colony may be formed by more than one cell. So you talk about it as colony forming unit, like if you were to make dilutions of a liquid culture and then plate that liquid culture on a solid plate and then get so many colonies on the plate. You can't say that there were 100 cells that formed 100 colonies on the plate, but you could say that there were 100 colony forming units that formed 100 colonies on the plate. Any question about that? And obtaining single colonies is a critical part of obtaining a pure culture. But remember, you have to streak it out twice for isolated colonies so that your cells go through two isolated colonies before we call it a pure culture. Even then, it's not 100% of the time going to be a pure culture, but it's greater than 95% of the time going to be a pure culture. And in microbiology, that is good enough. If over 95% of the colonies are a pure culture, we can call it a pure culture. Any question about any of that? So how you streak out to get isolated colonies is you just start from a stock culture, meaning a pure culture, and then let me scroll that up a little bit. Dip your, let's pretend this is a loop, dip it in the stock culture, we'll make that the tube, pick it out. Usually you get the worst of it off, and then start streaking it on sector 1. When you streak it on sector 1, this is actually a good one. Right here, it will be higher in numbers than down here. And you can maybe see a little bit about that. Right here, it's a little bit higher in cell concentration than there. And that's because as you're streaking, you are diluting it some. Not a lot, but you are diluting it some because there's going to be cells that leave the loop and enter the plate as you're streaking the plate. And then you take your loop and you sterilize it by putting it in the flame or in the Bacto incinerator, get it red hot. That kills any of the cells. And then you streak out quadrant 2. When you streak out quadrant 2, initially you will be going here and there's no cells on it. But then you go into quadrant 1 to pick up cells. You should go into quadrant 1 only 3 or 4 times. So run here, one there, and one there would be enough. And you're picking up cells from here and here and there in quadrant 1. This person did it a few too many times. But actually in quadrant 1, at least they didn't go where it was very high in density. What you want to do is go in quadrant 1 where the cells are reduced in number. And that would be there and there and there. The cells are reduced here, then up there. Any question about that? And then you pick up the cells here and then you start plating them on quadrant 2. I'm not sure. Well, this isn't the same streak here. So right there, they did the streak here, and it looks like they went here and there, and they went too far over here. You shouldn't go all that far. You should only go where, like about here, where the cells will be reduced. And that is actually shown for these. But like I said, they went too many times, and they shouldn't go that many times. You should only go 3 or 4 times into quadrant 1. And then you streak out quadrant 3, and this isn't quite shown correctly. It should be streaking here, coming here, and do it 3 or 4 times. This only has it 1 time, and that's not enough. Okay. Now here, where's quadrant 3? It's hard to see quadrant 3. This one actually looks okay. Assuming that they didn't go too far. They actually went too far because this goes all the way up here. They should only be going into quadrant 2 where the cells will be reduced, and that would be this region. You don't want to go up into this region where the cells are high in number. And then you streak out quadrant 3. And like I said, you should go more than 1 time. You should go 3 or 4 times into quadrant 2, but where the cells are reduced in number. And this is getting a little high, but this region would be good. And then streak out quadrant 3. And the idea is you're doing a dilution. First, there's a dilution here to there, and then there's a dilution between quadrant or sector 1 and sector 2 because you've sterilized the loop. So sector 2 will be much more reduced than sector 1. And that you can actually see here, very high numbers here. And you can see we're getting isolated colonies. They're a little too many, but we are starting to get isolated colonies in sector 2. And then sector 3 is even further reduced, and we're getting isolated colonies, a lot of them here. Any questions about that? Now, when you obtain cells from here and here, that may not be a pure culture because these colonies could be derived from more than one cell. So what you do is you take from an isolated culture and then you streak it out again on a plate to get isolated colonies. And then when you get your second plate, you take from an isolated colony, we'll take that one there, and then we grow that up. And that's gone through 2 colony forming events from the first plate and from the second plate. So these cells are likely to be pure, greater than 95% chance. Any question about any of that? All right, if there's no questions, let's move on. Drink that size. The growth of bacterial cultures. Remember that microbial growth refers to an increase in cell number, not an increase in cell size. The mechanisms by which bacteria grow are most bacteria grow by binary fission, where one cell simply splits into two. And we'll talk a little bit more about that in our next slide. But some bacteria can grow by budding, where they form a small outgrowth called the bud on the parent cell, and it enlarges with time, and it grows, the bud grows, and when it becomes close to the size of the parent cell, the bud will separate from the parent and then be its own cell. And there are a few species of bacteria that do grow by budding. Actually, budding is the way yeast grow by, and we'll look at how that happens. And then there are a few species, just a few species that reproduce by reproductive spores. You should know that a reproductive spore differs from an endospore. And a reproductive spore is reproduction because one parent cell will make it this big cell here. On the end of the cell form some reproductive spores, which are smaller. And the reproductive spores always show up on one side of the cell, usually the side exposed to the wind or a passing animal or something like that. Let me grow one more right there. Oops, a little short there. And you can see the parent cell puts out more than one reproductive spore. So totally here we have 5 different cells after the reproductive spores are made. So one cell ends up being 5 cells. That is reproduction. And what we're doing is these reproductive spores, the wind or a passing animal will pick it up and take that reproductive spore somewhere else where it will then grow into a vegetative cell, which will be similar to the parent cell, and it can grow elsewhere and then go through reproduction. And this species grows by reproductive spores, so it'll probably grow by more reproductive spores. Any question about any of that? All right, so reproductive spores differ from endospores, and I will try and always use the term reproductive spore or endospore when I'm talking about it. If you're talking about the word spore, and some authors do that, it gets very confusing as to what they mean. Do they mean an endospore or do they mean a reproductive spore? Remember, endospores are not reproduction. One cell forms one endospore, and then the parent cell dies. Endospores are a survival strategy. Reproductive spores are a reproductive strategy. All right, binary fission is shown here where the cell will replicate its chromosome, forming 2 DNA molecules, which will then move one DNA molecule to one side of the cell, the other to the other side of the cell, and then the cell literally splits down the middle. By literally, we mean that the cell does divide literally in 2, and it starts forming a cell membrane and a cell wall, which starts to divide the cell in 2. Here we're looking at an actual electron microscopic image of a cell that's going through division, cell division by binary fission. And that structure is going to be the cell membrane and the cell wall. And then it will form in between the 2 DNA molecules, dividing the cell into 2. Any question about any of that? And then the cell will then split and divide, assuming they're not going to be attached to each other, they will become two independent cells. Any question about binary fission? All right, here we're seeing yeast, and there's a yeast cell there, and there's the yeast cell that's putting out a bud, and there's the bud on a larger bud, and there's another larger bud. And these buds will grow until they become equal to the size or close to the size of the parent cell, in which case the bud will then come off and be its own independent cell. So let's watch a little movie of a bud or budding. There's the bud there, beginning to form. You can see the bud is growing, enlarging. And this is the DNA molecule there. You can actually see about now it's dividing into two. Let me back up a little bit. Here is where the DNA molecule is moving into the bud. So one molecule moves into the bud, the other stays within the parent cell. The bud then enlarges, and about there is where it separates from its parent cell. So it's its own cell now. And then as we go on, the parent cell is forming another bud here, and this is starting to form a bud there. And that's how budding happens. Any question? All right. Oops, I think I just stopped the sharing. Sorry about that. I didn't mean to do that. Meant to stop the video. Let's get rid of that. There we go. Any questions about budding? When we're talking about bacterial growth, we do use the term generation time. And as you know, the generation time for humans is, I don't know, something like 20, 21 years, something like that. It is the time required for one cell to divide into two cells, or for people, for two parents to come together to have one offspring. It's also the time required for the population to double in size. And then for humans, it would be for the population to double in size. And that's why I said the population doubling time, generation time for humans is something like around 21 years, which in first world countries, that's actually probably a little bit low, but we have a lot of third world countries, and they have a quicker generation time. And I would have to look it up to see what it is exactly. The generation time in bacteria varies considerably between different species. Some species can have as fast of a generation time as every 20 minutes. And we call the generation time 20 minutes, but realize that's the time it takes for the one cell to split into two, or for the population number from, let's say, 1 million to increase to 2 million. And we say the generation time is 20 minutes, but we mean 20 minutes per generation. But per generation is often left off. And E. coli has a very fast generation time. So its generation time is about 20 minutes per generation. Most bacteria have a generation time between 1 and 3 hours. But Microbacterium leprae the bacterium that causes leprosy, has a generation time of 10 days. So it takes this cell 10 days to go from one cell to two cells. Why the mycobacterium are much slower at dividing is because they have a lot of mycolic acid in their cell wall, and it takes them time to make that mycolic acid. Mycobacterium leprae is the smallest or the slowest bacteria I know of when it comes to its generation time. Now, when you talk about the generation time, remember it's always 20 minutes or a time per generation. It is never a time per generations per time. We're so used to thinking of miles per hour that we put the time second, but in the generation, it is time per generation. Is that clear? Anyone confused on that? And students often have trouble with the generation time. They think they know it, but they don't get the units right. Like I said, it is the time per generation. And E. coli can have a generation time of 20 minutes per generation. When we're talking about the generation time of bacteria, we like to use, in microbiologists, a semi-log plot, where we have the log of the number of cells on the y-axis, and then the x-axis is actually an arithmetic axis having the generation time. If we were to plot this with the cell number with arithmetic time on the y-axis and the time on the x-axis, which is also arithmetic, we would have a curve like this. We don't like curves like this because, let me blow this up. Scroll down here. I need to shrink that just a little. All right, one cell here divides into 2, divides into 4, divides into 8, divides into 16, divides into 32. So there's a whole bunch of cells or not points in the graph down here. And then you can't really separate them out on an arithmetic curve. And then the next point here, wherever it would be, whatever number it would be, I don't know what that would be, over 2 million, it'd be close to 2,100,000. Where that will be, you can't put it on the graph here. It's going to be on the ceiling because this is an arithmetic curve and it's exponential growth. So this distance increases exponentially. So there's no way to get the next point on the graph. Whereas with the semi-log point, these points are separated better down here. I don't know how to word it, but it's just that they are separated more. There's more space between. Well, it shows you here. Here's 1 and 2, for example, that there's a lot of space. That's not 1 cell and 2 cell. That's log 1 and log 2 are separated. So they're separated out more. You can easily plot them in. And then the next point can actually go on the graph. It's going to be about right here. So you just extend this graph and then you can put the point out there. And then the real reason why microbiologists like the semi-log plot is this equation is very easy. It's y equals mx plus b, a very simple mathematical equation. Whereas this one is a very complex equation. It's going to be something like y equals mx plus b plus mx squared. And there might be another constant in there too. Okay. So mathematicians and microbiologists like to use a semi-log plot. When we're talking about bacterial growth curves, we actually get almost always a growth curve looking like this. And this is not unique to bacteria. This is a growth curve you tend to find for almost any organism. Like somebody introduced a deer population to an island, I think it was on the East Coast, and the deer population followed this curve. So initially, when the deer were moved to the island, they were disturbed, so they had a lag phase. And then they were on a new place, they had to explore it before they started getting interested in each other and started reproducing. So there was a lag phase. And then they started reproducing, and there was only a few deer on the island and lots of food. So the deer population grew exponentially. And then the deer population plateaued. That's because now there were lots of deer on the island and not very much food. So the deer population numbers had some growing, but those growing were equal to those dying because there was not enough food for everyone. And then we reached the death or decline phase where the deer are not growing their population number because there's not enough food, so they're all beginning to die. And eventually they would all die on the island because there wasn't any more food for them. Any questions about any of that? For bacteria, that we follow the same growth curve, and that is when we move bacteria from like a agar plate to a liquid media, there's a lag phase. The bacteria are not growing initially when we inoculate them into a new media. What the bacteria are doing here is they're getting their enzymes ready to grow in the new media. So they are metabolically active, but they are not ready to grow in the new media because they don't have the enzymes for growing in the new media. And like I said, they were in a solid media and we moved them into a liquid media, and that's why there's a lag phase. Once their enzymes become ready for growing in the new media, they then enter an exponential growth phase where there's few cells in the media, lots of nutrients, lots of space. However, as the population number grows, we will find that there will be less nutrients and less space for the cells. So their growth will start to slow. And they will eventually reach a stationary phase where the lucky cells have some nutrients around them, they have some space around them, and they don't have that many waste products around them. So these lucky cells are growing in the stationary phase. But the unlucky cells have lots of waste around them, very few nutrients, very little space, and they're dying. So in the stationary phase, the number of cells growing is equal to the number of cells dying. And that will continue for a while. And then the death or decline phase happens where there's more cells dying than there are cells growing. And that's only in the early death or decline phase. Late in the death decline phase, there's no cells growing. The cells are only dying. That's because there's no more nutrients around, very little space, and there's lots of waste products around. And the waste products are inhibiting the growth or killing the cells. And obviously they can't grow because there aren't nutrients around. How quickly the population will die off will depend on the cells. E. coli, for example, will fairly quickly die off within days, like about 2 weeks. after setting up the culture, probably most of the cells, if not all the cells of E. coli will be dead. However, if the culture makes endospores, the cells can live for decades or millennia. We know this because, like on Egyptian mummies, we've germinated some endospores. And as you know, that's thousands of years ago. I used to know how long, something like 6,000 to 8,000 years ago. And the endospores can still germinate. The longest they have is something like 40,000 years, or maybe it's even longer. Maybe it's millions of years, and I got my numbers wrong, where they took a ice core sample from Antarctica, and they found endospores in the ice. And then they thawed it out and then had the endospore germinate, and it still survived. So like I said, how long the death or decline phase will depend on how quickly the cells die. Most gram-negatives, like E. coli, don't live that long. Gram-positives tend to live a little longer, but even Staphylococcus aureus will eventually die after a few weeks. This doesn't go on for years. But if it's endospores, that can go on for not more than years. It can go on for decades, millennia. Any question about any of that? All right, if not, let's talk about the different ways that cells can grow and how we can measure their growth. There are different methods we have for measuring the growth of microbial populations. There are direct methods and there are indirect methods. Direct methods include a plate count, also called the viable plate count, filtration, most probable number, direct microscopic count, where you're counting cells under the microscope, turbidity, metabolic activity, and dry weight. I've used all of these methods for counting microbes except for the most probable number. I've never used the most probable number for making a cell count. In a viable plate count, also called the plate count, it's the most common method used in microbiology for measuring a population number of cells. It does measure the number of viable cells, and that's why it's sometimes called a viable plate count, because the only cells you're counting are viable cells, because a viable cell will go on to form a colony, and that's what we're counting, colonies. A non-viable cell will not grow into a colony. Because we do not get the exact cell number, because a colony can be formed by more than one cell, we often report the results as colony forming units per mil rather than cells per mil. Is that clear? Because when we have a dilution from our sample and then get so many colonies from that dilution, and then we estimate the number of cells per mil, we don't know the exact number of cells because some of the colonies are formed by more than one cell. So we count them as colony forming units per mil. As I stated, the plate count or viable plate count is the most common method used in microbiology. It starts by making a serial dilution of your original sample. So here we have our original sample, and the bacteria numbers would be too high in the original sample. So we will take 1 mil of that and put it in a 9, and now we have a dilution of 110. we mix this tube up, take 1 mil of it, put it into 9 mils, and then we have another 1 to 10 dilution for a total of 1 to 100 dilution. Mix those cells up, take 1 mil out, put it into 9, and then we have our 1 to 1,000 dilution, and then do that again, 1 to 10,000 dilution, do that again, 1 to 100,000 dilution. And then for each of these dilutions, you mix the cells up in the tube, and then you plate them. There's 2 ways you can plate them. You can just pour 1 mil onto the plate and then move that media around on the plate, allow the media to be absorbed, and then incubate the plate upside down for a period of time, a day or 2, and then bring the plate out, and then you'll see that it has colonies on it. And then you can count those colonies and then obtain the colony forming units in that dilution. And that's the pour plate method. There's also the spread plate method, where you put a smaller volume down of media from the dilution, and that could be 0.1 mil. And then you need to spread that around with what is that called? The spreader. So that media is spread on the plate, and then you incubate it, and then count the colony forming units forming the number of colonies on the plate. Any question about that? Now, some of the dilutions will have too high of cell numbers to count, in which case you report the 1 in 10 dilution as TNTC. Is that shown there? Too numerous to count. Also too numerous to count. Still too numerous to count, although you could get an estimate on that one. And then on this one, we can count the number of colonies there. And that's going to be around 30 colonies. And this one, we can count the number of colonies here too. It's going to be around 4 colonies. So which plate do we use to get our estimate? This plate or that plate? Any ideas?
The one with 4?
The 4 is easier, but we actually don't use this one. We use that one, and the reason why, let me make a new plate here. Let me blow this up so you can see it. So on the 4, if we have an error of plus or minus 1, let me go up there. Go back to that plus, make it a plus or minus. There we go, plus or minus 1. Actually, let's do 3 just to make it easier. That means that the variation there, that's plus or minus. Let me copy that there. That's plus or minus 33%. That's a very high, whoops. Oh, crud. It's a very high error estimate. On the other hand, if we have 30 colonies, plus or minus 1, it's not plus or minus 3. I think it's plus or minus, and hopefully I have my math right here. Is that right? 1 out of 30 is going to be about 3.3%. That sounds about right. This is an error, smaller error number. So we like to use this mean because this mean has a smaller error. So this mean is more correct. This one has a very large error. And that's only off by one colony, 33%. So we like to use this one that gives us a better estimate of our mean. Any question about any of that? Oops, what am I doing here? So we want to make the colony estimate based on this plate because the mean means something. On this one, plus or minus 1 is just too large of an error. Anyways, that's the viable plate count method. It only counts viable cells because a non-viable cell will not go on to form a colony.
So I understand that the percentage is smaller with the 4 than
Uh-huh.
it is the 30, but so it's 29 or 31 on the more that could be viable.
I'm saying that the mean here actually means something. If we say 30 plus or minus 1 is not that big of a difference. Here, where it's 4 plus or minus 1 is a big difference. And here it would be like 25%, plus or minus 25%. Okay? So we want to use this one because the estimate of the mean is better. Any questions? All right, let's move on to the filtration method. Filtration is used when the number of cells per mil is very low, like when we're assaying drinking water for the number of bacteria in it. There will be very few bacteria in it. And so if you put 1 mil on a plate, we get very few colonies. We might not even get any colonies growing on the plate. And so obviously the bacteria number is low. And so you don't want to make a serial dilution. What you want to do is increase the cells to a concentration where then we can make a colony count. What you do is you take the liquid or maybe a gas, and then you send it through a filter. And here we're seeing a filter here. And then the cells get caught in the filter. And then you take the filter and then you put it on a plate of media. And then you grow up the colonies that were trapped on the filter. And here, I don't know, 50 to 100 colonies are actually growing here. And you'd have to say something like, let's just say 50 colonies per however much you sent through the filter. It could be 50 colonies per gallon of water, which I suppose that would be appropriate. I'm guessing that a gallon of drinking water might have 50 colonies. I mean, I don't know because I know it's very low though in our drinking water. And that's why you would send it through a filter. Any questions about that? So filtration is similar to a viable plate count. The difference is you increase the concentration of cells on the filter and then grow the filter on the agar. All right, let's move on to the third way we can directly measure microbial growth, and that is by a direct microscopic count, where you count the number of cells under a microscope. You usually use a gridded slide that has a specific volume under it, and then the cells are counted microscopically. It's not good for motile bacteria. I'll explain why in a minute. It does count both viable live cells and dead cells. It does require a high cell concentration, but you can centrifuge the cells to increase the concentration of cells. No incubation time is required. That's one advantage of this, but it does take someone to count it. So for the poor schmuck who's got to count it, it's actually a little tedious, but it's pretty quick too. Like I said, it's usually done on a gridded slide. The slide here has a specific volume under the slide, and it has a grid on it. The grid is usually such that one such square here has a volume of 1 microliter. Is that right? And then you take the cell number here that is obtained, I don't know, 15 cells there, and then you multiply by 1,000,000 to get the number of cells in a mil of your original sample. So this volume here underneath this grid right there is very slight. And then so you take your cell counts and you multiply by 1,000,000. That's usually the way a gridded slide works. And there you can see the slide with a cover slip, and the slide is designed so that there will be a specific volume underneath that slide. And it's even designed so that under this grid, it's a specific volume of something like 1 microliter. The problem with motile cells is if you once count the cells and then these move in and then these move out because they're motile, it's very difficult counting motile cells because like these two could move over here and then you could miss them. And so it's very difficult counting motile cells. What you could do is kill the cells and then do your cell count. It's a little difficult counting cells if they have capsules because they just will be much larger here, and it just makes it harder to count. Like this one, the capsule will be partly in the slide and will be partly out. This one would definitely be in the slide that had a capsule and partly out. And so how do you count them? But capsules can be counted. They're just a little more difficult than plain cells. All right, I'm not going to talk about the most probable number way because for one thing, I've never used it and it's a little more complex too. And you guys will probably never use the most probable number for counting cells. The indirect methods, we'll talk about how you can indirectly get your number of cells per mil for your microbes using turbidity, metabolic activity, or dry weight. Turbidity is one of my favorite ways to count the bacterial numbers. It's an indirect method measuring the amount of light coming through the tube. So here we have a tube of media, and so much light comes through that. You then compare it to this tube that had the media and no cells in it to a tube that has cells in it. Let me blow this up a little. Move that over. Wherever you have the cells, it'll block the light getting through. So less light will get through this tube compared to that tube. And then you go look at a chart. Somebody had to make the measurements and then record the amount of light for how many cells you have. So somebody has made up the chart, but once you have the chart made, you can use that chart for years. Okay. And then you just plop in a tube of media, plop in a tube of cells, get your number of light coming through, look at the chart, and it gives you the estimate for the number of cells per the amount of light coming through. Okay. A spectrophotometer is just a device that measures light. And so you need a spectrophotometer to do that, turbidity meter, but it's a very quick way of doing it. You just take your media and take your cells in the media, put it in the turbidity, measure the light, look at the chart, and then you see how many cells you have. Any question about any of that? So this is the quickest method. A metabolic activity is where you're measuring something like acid being produced or CO2 being produced, and you make the assumption that you get more CO2 or acid if you have more cells present. And then you measure the amount of like CO2 being produced, compare it to a chart, and then that gives you a method for obtaining the number of cells. Somebody has to go through and measure the amount of CO2 being produced, usually in a given minute of time. So you, like for CO2 being produced, you allow the cells to start producing CO2 for something like 1 minute or 10 minutes. And then you measure the amount of CO2, and then you look at the chart and you see that, let's say it's CO2 is like 32 units of CO2. And then you look at the chart and you say, how many cells? Let's say it's 1,000,000 or 32. And then that's your estimate. Somebody has to go through and make the chart, but once you have the chart made, it can be used for years. Any questions about metabolic activity? I used to have students in the preliminary class measuring the amount of CO2 being produced, but I don't know if that's still done. I haven't taught the preliminary class for years now. And if you took it online, obviously you wouldn't have done that. The dry weight is another method used for measuring bacteria and get an estimate. It is a usual method for filamentous bacteria and molds, where what you do is you grow up your cells and then centrifuge them down, pour off the liquid, and then dry the cells. And then once they're dry, you then measure the dry weight. And then you compare it to a chart for the amount of weight or the number of cells you have. Somebody has to make the chart initially, but once you have the chart, you can use that chart for years. Some advantage for the dry weight method is once you have the cells dried down, at your own convenience, you can measure how heavy the cells are. So it doesn't have to be done right away. You obviously have to grow the cells, centrifuge them down, and dry them. But once they're dry, you can then measure the weight whenever you want. It's not a thing. It does have the disadvantage, in fact, all of them, except for the viable plate count infiltration, will measure the number of dead cells. A dead cell will add weight to the dry weight. A dead cell will block the light in turbidity. A dead cell, when you're looking at it under the microscope in the cell count, let's go above. You can't tell, is that cell viable or is that cell dead? So you just count the total number of cells, and only both the non-viable and the viable cells will be counted, except for the viable plate count. Okay. All right. Any questions about the different methods we have for obtaining an estimate of the number of cells? If not, that's it for this lesson. Let me see where we are. And if we, we're supposed to start chapter 8 today. So if there's no questions, we're done with chapter 6. Let me begin chapter 8. Got two files here. I'm assuming it's the more recent one, but let's take a look there. See if that has all of them in there. Yep, that has all of them. All right, chapter 8. This slide is giving you the major goals and a rough lesson outline. Know the terms. What's a genome? What's a gene? What's a genotype? What's a phenotype? What's vertical and horizontal gene transfer? What's an operon? What is a promoter? What is an operator? What's a structural gene? What is a crossing over? What is transformation, conjugation, and transduction? 2, be able to describe DNA replication, RNA transcription, and protein translation. And then 3, understand the different types of mutations. Any questions about what we'll be talking about? This is a summary of what we're going to be doing. We'll talk about the structure and function of the genetic material, talking about the flow of genetic information, how the information in the genes flows in the cells. We'll talk about DNA replication, RNA transcription, and protein translation. We'll then talk about the regulation of bacterial gene expression, using the operons to discuss that, and talk about, then we'll talk about mutation, we'll talk about genetic transfer and recombination, and then we'll end the lesson on talking about genes and evolution. Any questions about what we're going to be discussing? Not hearing anything, so let's move on. Oops, what happened here? Genetics is the study of what genes are, how they carry information, how that information is expressed, and how genes are replicated. A gene is a segment of DNA that codes for something. Usually, it codes for a protein, but not all genes code for protein. Some genes only code for RNA, and that would be like the tRNA gene or the rRNA gene. They only code for RNA. Any questions about any of that? So a gene is something that codes for something, usually a protein, but it doesn't have to be a protein. It could be RNA, it could be tRNA, rRNA, or some other form of RNA, which we're not going to talk about. Here we're looking at a chromosome. You'll note that there is a gene here and a gene there, and that would be a non-coding region right there in between gene 1 and gene 2. And gene 1 would be there on the chromosome, gene 2 would be there. And there we're seeing a human chromosome with the different locations of different genes. Any question about any of that? All right, not hearing anything, so let's move on. A genome is all of the genetic material within a cell. For microbes, the genome is also all of the genetic information within the organism. A genotype is the genes of an organism. For example, a blue-eyed individual has 2 lowercase b genes, and we'll call those alleles. Let me get this out of the way. So the blue-eyed individual has 2 lowercase b's. A brown-eyed individual has 2 genes, but they may be either lowercase b, uppercase b, and I guess I should say uppercase b, lowercase b, or 2 uppercase b, uppercase b. The uppercase b allele stands for brown, and it dominates. We'll talk a little bit more about that later. And that's why the brown-eyed individual can be either way, where they have an uppercase B brown-eyed allele and maybe a blue-eyed allele, which is recessive, or they can have 2 brown-eyed alleles. Anyways, that's their genotype. The phenotype is the expression of the genes in an individual. For the blue-eyed individual, the expression of the lowercase b genotype is blue-eyed. For the brown-eyed individuals, the expression of the genes is brown eyes, and they can have the genotype either capital B, lowercase b, or capital B, capital B. Any question about that? So in simple genetics, the phenotype is determined by the genotype. That is only the case for simple genetics. What's the term? In polygenetics or complex genetics, this simple relationship does not happen. And something like a person's weight would be a complex genetic genotype where there will be more than one gene that can affect the person's weight, but then the person can also have more or less weight on their environment, meaning if they're constantly eating, they're going to be heavier than if they're, constantly dieting, for example, or they're eating very little food. They'll weigh less. And the point is, weight is a complex trait. It is determined both by the genes, and there's many different genes that can affect the weight. When I'm talking about, the genes can be at different locations and in the different chromosomes. So there's many different genes that can affect the weight. And then the environment can affect the weight as well. We won't talk about complex genetics in this class other than here, but we'll only talk about simple genetics in this class. Any question about any of that? All right, here we're seeing the E. coli chromosome where the DNA is on the chromosome, and E. coli only has one chromosome, or at least oftentimes it only has one chromosome. And it's just a string having one gene after another on it. Remember, a chromosome is both DNA and protein. And for the moment, we're only talking about the DNA portion. DNA is a polymer of nucleotides, and DNA can have 4 nucleotides: adenine, thymine, cytosine, and guanine, or you can call them A, capital T, capital C, capital G. You must know one or the other, but I don't care which. Any question about any of that? Okay. DNA is a double helix here and here, meaning it has two DNA strands. Each one single strand of DNA, let me blow this up, is held together by the sugar, deoxyribose, phosphate backbone. So the one strand of DNA is held together by the sugar phosphate backbone holding the DNA together. And that's a covalent linkage. The two strands of DNA are held together only by hydrogen bonds between the bases, the nucleotide, the nitrogen containing bases. A and T have two hydrogen bonds between them. G and C have three hydrogen bonds between them. So the two strands of DNA are only held together by hydrogen bonds. Each of these hydrogen bonds is weak and will break, at least at our body temperature, all the time. But the thing is, when this bond breaks, that one and these are holding the DNA together, and then this one can reform, and then that one can break. But once again, this one is holding the DNA together, and those are holding the DNA together. Any question about any of that? All right, I also need to say that DNA is anti-parallel. That means one strand of DNA is running this way. Let me go like that. And the other strand of DNA is running that way. So one strand has the 3' end over here, the 5' end over here, and the other runs in the opposite way, where the 3' end is over here and the 5' end is over here. You do not need to know this, but the 3' and the 5' refer to the carbon in the sugar. This carbon here is the 3' carbon, or the third carbon. This carbon here is the 5th carbon. And right there, that would be the 5th carbon. And that's why they say this is the 3' end and not the 5' end. You don't need to know that. I'm just pointing out where the numbers come from. And it's just the way that chemists discuss sugars, okay? You should know that the strands of DNA are anti-parallel, where one strand is running that way, the other strand is running that way. And let me undo that. All right, the DNA encodes information within it, and that information will flow within the cell in a certain direction. The information within the DNA flows from the DNA to the RNA in transcription, and then that would be from messenger RNA to protein in protein translation. So this is the vertical flow of information within a cell, from DNA to RNA to protein. This is known as the central dogma in biology, which is a law in biology, because in our cells, the information always flows, at least from our genes, from DNA to RNA to protein. It never flows like from protein back to RNA, and then RNA to DNA. The central dogma happens only in our cells. For example, in viruses, information can move from RNA to DNA. So the central dogma happens in our cells, but in retroviruses, the information actually flows from RNA to DNA. And we'll talk about more about that later when we're talking about retroviruses. Any question about any of that? All right, so this is showing you how the information of DNA flows in a population of cells. Within a cell, the DNA only flows from DNA to messenger RNA to protein. And that we've already talked about. But between generations of cells, the information can move from the parent cell to the information in the daughter cells. Or for humans, the DNA, information from the DNA can move from two parents to the DNA in the offspring. And that we call the flow of information between generations of cells, or in people, it's the information of flowing between generations. Any question about any of that? We also have the flow of information flowing from one cell, a donor cell, to a recipient cell. And you'll note that I've broken this slide here and added that information there. The DNA comes out of this donor cell and then goes into the environment. And then the DNA is picked up by the recipient cell that we call a recombinant cell. The DNA flowing out of this cell usually kills this cell. So this cell usually dies. Doesn't always have to, but usually, or at least often, it dies. And then the DNA is gained by this cell. This we call the flow of information between cells of the same generation. Any question about any of that? When we're talking about the vertical flow of information, where DNA flows within a cell from DNA to RNA to protein, the information is stored in a language of DNA that we call nucleotides. And there are 4 different nucleotides in the alphabet of the language of DNA. That information is then transcribed into the information stored in RNA. And once again, that language is nucleotides. But these nucleotides are a little different because these aren't DNA nucleotides, these are RNA nucleotides. And once again, there are 4 RNA nucleotides, so that in RNA, there are only 4 letters in the alphabet of RNA. And then the information moves or flows from the RNA to the protein, and the language is the language of amino acids. And there's 20 different amino acids in proteins. And so the information is now stored in the amino acids, and there's 20 different of them. A comparison, English has 26 letters in its alphabet. Any question about any of that? When we're looking at the information within DNA, we could say that the information codes for that is a dog. When the information is transferred to the RNA, we have the same information, but it is a different dialect because it's stored in the RNA nucleotides, not the DNA nucleotides. So instead of saying that is a dog, you'd say something like, that is a dog. Same information, just a different dialect. But when it's moved into the amino acids, it's stored in a totally different language. It's the same information, but in a different language. And it would be something like, quello è un cane. If you don't know, cane is some language for dog. It might be Italian, I don't know. Un is a, A apparently as quello is that. Any question about the vertical flow of information within cells? If not, let's talk about the, vertical flow of information within a cell. Let's talk about the flow of information between generations of cells, and this is also a vertical flow of information. The information flows from the parent cell to the daughter cell. How that happens is the information in the DNA is copied, so the DNA is replicated, and then we have two strands of DNA. One will move towards one side of the cell, the other to the other side of the cell, and then the cell will divide in the middle so that each daughter cell gets the same information in the parent cell from their chromosome. That's the vertical flow of information within, between generations of cells. How that happens is when DNA is replicated. Let me see if I can blow this slide up. So when DNA is replicated, we separate the two strands, and then one strand is used as a template to make the newly growing strand. So this has a T here, so the polymerase will put an A here. This has a G there, so the polymerase will add a C. This is a C here, so the polymerase will add a G. This we say is DNA replication happening semi-conservatively, where one strand is used as a template to build the new strand. And then that's true for this strand here. One strand is used as a template to build the new strand. That's called semi-conservative replication. Come on. So that when DNA replicates, the black being the old strand, the original strand, each daughter cell will have one black original strand and one new strand. Same for the other daughter cell. That's different from conservative replication, where one daughter strand would have had two black strands, and then the other daughter strand would have two white or new strands. Conservative replication does not happen. It's a semi-conservative DNA replication that happens. And don't ask me what liberal replication of DNA would look like, because I don't know. Any questions about any of that? So DNA replication just uses one strand as a template to make the other strand. So this C is a C here, it'll add a G. This C is an A here, it'll add a T. And this is trying to show you that nucleotide triphosphates are added. And then this bond here is broken, and that's a high energy bond that releases the energy needed for this T to be added to this newly synthesized strand of DNA. And that will be a covalent linkage between the phosphate here and the sugar here. Any questions about that? Which is, I guess, shown here. So there's the sugar, phosphate, sugar, phosphate, sugar, phosphate, holding one strand of DNA together. Any question about any of that? All right, if not, let's watch a little video on DNA replication.
Communicate in English easily. Use Grammarly and feel confident with effective
Like commercial.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions about this movie? Move on. So DNA is copied by DNA polymerase. Oops. Lost my place. The new DNA is always transcribed in the direction 5' to 3' direction. It is initiated by an RNA primer, but I'm not going to test you on that. The leading strand is synthesized continuously because it's running in the right direction, 5' to 3'. So this new strand, once that DNA polymerase is made, will just keep running down that strand. The other strand is the lagging strand, and it cannot be synthesized directly because it is running in the wrong direction. Let me see if I can blow this up. So it has to be made in the 5' to 3' direction. And instead of being made this way, the DNA polymerase looks like it started here and then moved this way. And then this one started here and then it's going to move this way and we'll go to there. And then that one started here and is going to move there, and we'll go to here. And this one will start here and go to there. Wherever the DNA polymerase adds its first nucleotide, let's say this one's going to add it right here, there will be a gap where this new strand of DNA is not bound to this strand of DNA. And here, we're going to have lots of gaps. There's going to be one between this strand of DNA and this short piece that's called an Okazaki fragment. And then there will also be one between this short piece and this one. And then between this one and that one, etc. So there will be lots of gaps in the lagging strand of DNA, where the new strand of DNA will have different breaks in it. It will not be connected. This Okazaki fragment is not connected to this strand of DNA. And on the leading strand, it'll only be where the polymerase first starts. So right here, let's say it starts right there. This strand of DNA will not be connected to this one. How that is connected is the enzyme ligase comes in and will put, I guess that's what that blue one is. I didn't realize that. We'll connect this piece of DNA to that one. And in this strand, the ligase only works wherever the polymerase first began, and it will connect this newly made strand, which isn't shown here, but it'll be right there. We'll connect it to here. The point is that ligase doesn't work very often in the leading strand, only where the DNA polymerase begins. And if it begins at the very start, and then that DNA polymerase were to transcribe this in whole chromosome, the ligase would never work. But usually the DNA polymerase will begin and then fall off, and then another one will begin here and then move on. So usually the leading strand does have one or two or a few breaks, but the lagging strand has many breaks. There will be a break between each Okazaki fragment. And then between the new strand, which actually that's, we'll just call it the new strand. That might be just an Okazaki fragment, but it could be a new strand. And the first Okazaki fragment. So the ligase works quite a bit on the lagging strand. And the ligase is making a covalent linkage between this nucleotide here. Come on. Sorry, my mouse is dead. There it goes. It's making a covalent linkage between this nucleotide here and then this nucleotide right there of this strand of DNA, or between this Okazaki fragment, let's say here, and then this one here. So it's making a covalent linkage between the pieces of DNA. Any question about any of that? So the important enzymes to know are the DNA polymerase that transcribes the new DNA, the helicase, which opens up the two strands of DNA and makes them single strand. You can only replicate a single strand of DNA, but the helicase opens up the two strands of DNA and makes it single-stranded. And then the DNA polymerase makes the new strand of DNA, and then the ligase is important at linking the new strands of DNA or the Okazaki fragments together. Any question about any of that? I think that's it. The leading strand is synthesized continuously. The lagging strand is synthesized discontinuously, resulting in many Okazaki fragments. And then the DNA is put together by DNA ligase. All right, so that's DNA replication. let's talk briefly about RNA transcription. I think we've got time. Let me check on that. I think we go to 6:50. Yeah. I guess I should talk about whether we have a lab today, now that I think about it. We do have a lab today, lab 8. Why do I have 2 of those open? So let's talk about RNA transcription. Transcription is the synthesis of RNA from DNA. The DNA is transcribed to make RNA. The RNA can be either messenger RNA, tRNA, or rRNA. Here's a question for you. How does the RNA polymerase know whether to make messenger RNA, tRNA, or rRNA from the DNA that it is transcribing? Anyone have a guess?
It codes for it.
Yeah, it depends on what gene the RNA polymerase is transcribing. If the gene codes for a protein, it will transcribe the RNA as messenger RNA. If the gene codes for tRNA, the RNA polymerase will transcribe the gene as tRNA. And then the rRNA gene will be transcribed as rRNA. So the RNA polymerase doesn't need to know which RNA to make. It just transcribes the gene, and the gene itself will code for which RNA will be coded. And what's the difference between messenger RNA, tRNA, and rRNA? Are there any difference in nucleotides? That's either a yes or no question. You have a 50-50 shot at it, even if you're just guessing. And you should know this one, by the way. Are there any differences in the nucleotides, messenger RNA, tRNA, transfer RNA, or ribosomal RNA?
No.
Yeah, they're the same nucleotides. The only difference between the RNA of messenger RNA, tRNA, and rRNA is the sequence of the nucleotides. If it's a tRNA, it'll have a specific sequence of nucleotides. If it's an rRNA, it'll have a specific sequence of nucleotides. And if it's a messenger RNA, it'll have a different sequence. And the sequence of messenger RNA will vary because there are so many different genes that code for a protein. But there's only a handful of genes that code for tRNA and a handful of genes that code for rRNA. Any questions about any of that? All right. Transcription begins when the RNA polymerase binds to the DNA, and the RNA polymerase binds to the promoter. Let me draw that if I can. So here is our DNA. And which region of the DNA will be transcribed? Or another question would be, an easier question is, will all regions of the DNA be transcribed? Yes or no? Let's talk about a eukaryotic genome. Will all the eukaryotic DNA be transcribed? I'm not hearing anything, so I'm trying to
Just chunks. Fragment.
chunks. Yes, that's correct. Only the genes will be transcribed. So that's gene 1. See if I can get this in here. Probably not. Let's drink that up a little. Might be able to get that up. There we go. So only the genes will be transcribed. None of this region in between the genes that doesn't code for information will be transcribed. And remember, on eukaryotes, most of the DNA is non-coding DNA. And prokaryotes, most of the DNA is coding DNA. So most of the DNA does code for a gene in prokaryotes, but in eukaryotes, it's the opposite. Most of the DNA is non-coding DNA. And what does that non-coding DNA do? It's not entirely certain what all the non-coding DNA does. Obviously, it does hold the DNA together, but the non-coding DNA probably is important in the folding of the DNA to form the chromosome. So the non-coding DNA is important in the folding of that DNA to get it to form the chromosomes. And if you remember, in mitosis and in meiosis, the DNA will be highly folded up to make the visible chromosomes. But other than in mitosis and meiosis, the DNA will not be folded up and will be dissolved in solution in the nucleoplasm of the nucleus. So the DNA will actually be in solution. But when it is in a visible chromosome, the DNA is obviously folded up and will precipitate out of solution. So that's something that what the non-coding DNA does. It does serve at least a couple of purposes, and that's one of the major ones. Okay? So only the genes are transcribed, and when the RNA polymerase binds to the DNA, it will bind at the promoter. Oh, I got the wrong thing here. The promoter is a region of the DNA upstream of the gene. And usually it's very close to the gene. I sort of drew that away from it. I meant to be a little closer. Let's try and make it closer. I can bring it here. There we go. Get the eraser in there. And that we're just going to put a P for the promoter. The promoter, what its purpose is it codes for the binding of the RNA polymerase to bind here. And then the RNA polymerase will bind here and then will move down the DNA, transcribing it. And so, yes, a little bit of DNA is transcribed before the gene, but it's only a little bit. I don't know how many nucleotides, but it's not very much. And then it'll transcribe the gene, and then that will go into the cytoplasm of a eukaryotic cell and will then be used for protein translation, because that'll be transcribed as messenger RNA, because this gene, we're going to say, codes for a protein. But anyways, the promoter is a region of DNA upstream of the gene, and it's very close to the gene, and it codes for the RNA polymerase to bind to the gene. Any question about any of that? I guess I'll leave that open just for a little bit. All right, and then the RNA polymerase moves in the 5 to 3' direction. So RNA is made in the 5 to 3' direction, being transcribed. Let me go to my picture again. So RNA will be made about here. And this RNA will actually hydrogen bond to the single strand of DNA. I didn't mention it, but transcription and replication only happens on single-stranded DNA. So you do have to open up this DNA with a helicase before it can be transcribed. And then the RNA polymerase binds the promoter, begins transcribing the DNA, making RNA. And then at the end of the gene, I'm going to put that right there. Let's make it a little bigger so I can put in the T. Let's see if I can get that. Right here is the T. Up. See if I can move that over. Let's erase that. Up. Ah, you can sort of see the T now. The T is the terminator. The terminator is a region of DNA downstream of the gene, and it's right at the end of that, gene 1. And the terminator codes for the RNA polymerase to fall off the DNA. So once the RNA polymerase reaches the T, the terminator, the RNA polymerase will fall off. And let me try and draw the RNA polymerase. We're going to make that a circle, being the RNA polymerase. It'll fall off the DNA. That will release the RNA, and then that will be a free strand of RNA. It'll no longer bind to the DNA, and because that's only a hydrogen bond, and it will then move into the cytoplasm for a eukaryotic cell. In a prokaryotic cell, this is already in the cytoplasm. I think that's it. So transcription stops when the RNA polymerase reaches the terminator. The terminator codes for the RNA polymerase to fall off the DNA. Here we're actually looking at a microscopic picture. It's an electron microscope. It's not a transmission electron microscope or a scanning electron microscope. This is actually a fluorescent electron microscopic image. And there you can see this is the DNA, and that's RNA polymerase on the DNA. And right there, we're seeing a little bit of RNA being transcribed. Any questions about any of that? Let's go through.
It's
Actually,
6:50.
it's past time, so let's stop here. What?
Oh, I was just reminding you of 6:50, but you just said it, so you're
Yeah,
straight,
okay.
man.
All right, thank you. All right, I'm a little past 6:52, so let's say 7:02. I'll begin the lecture. And the reason why I want a little bit of extra time is I've got to go run to the bathroom, and then I'll begin the lab at 7:02. Any questions before I go? All right, see you in the lab.